Hear me. Hi everyone. Again, since the recording started, I hope you can hear me. I'm not sure if you can see me. For some reason, I can't turn on my video, but that's okay. The slides are the important part here.
I'll try to see if I can work on that from the back end. I don't know why.
No problem. I'll try a few more times, but in the meantime, I'll start talking about Descartes-8 and Cartesian Therapeutics. So I'm Miljković. I'm the Chief Medical Officer at Cartesian. I'm a hematologist oncologist by training, but as you'll see, most of my work has been dedicated to neurology in general and myasthenia gravis in particular. So I hope tonight that you will learn more about chimeric antigen receptor or CAR-T cells. I'll say a few words about why we think that RNA CAR-Ts are more appropriate than conventional DNA-based CAR-Ts for treatment of diseases other than cancer. And I'll say a few words about how our CAR-Ts are now being evaluated in myasthenia through different phases of clinical trials. So myasthenia gravis, as you know, is an autoimmune disease. It's a disorder of the immune system. And at the same time, we're using CAR-T cells, which are an immunotherapy. So we're using the immune system to combat an autoimmune disease. And the immune system in general is very complex. And people have tried different ways to show all of the different actors, all of the different cells that are in play. This is one of those ways. This is an illustrator who for Rice University drew nice cartoons of different cells that each plays an important role in a healthy immune system and in how the immune system fights infections, most of all, bacteria, viruses, parasites. For our story, we don't need all of these 12 actors. We are focused on two. We are focused on cytotoxic T cells and on plasma cells. And in healthy immune systems, both of them serve an important role. Cytotoxic T cells are there to combat mainly viruses, but also cancer. So they are a normal cancer surveillance mechanism. And plasma cells, their main role is to produce antibodies. And antibodies are our body's protection against viruses, against bacteria. And you probably heard about, especially COVID antibodies that are a result of either a natural infection or of a vaccine. However, in autoimmune diseases, these cells can go rogue. Plasma cells in particular, they can produce antibodies that are not directed against a virus. They're not directed against COVID. They might be directed against our own bodies, parts of our own bodies. And in myasthenia, those antibodies are directed against something called the neuromuscular junction, which then results in the weakness that's associated with myasthenia gravis. The cytotoxic T cells got their name from their role. So cyto is cell, toxic is toxic, deadly. Its role is to kill other cells. Here you see a T cell in yellow engulfing a cancer cell in blue. And you can see that when it plays its job, it's engaging another cell and it kills it. So it's very good at what it does. The trouble is it needs a lot of support. So these are the other supporting cells that cytotoxic T cell needs to know which cell to target. Here is an antigen presenting cell. Here's a macrophage. Here's a T follicular helper cell. So they usually are there to prevent the cytotoxic T cell from hitting an unintended target, because sometimes even cytotoxic T cell can cause autoimmune disorders. What CARs do is short circuit and bypass all of these mechanisms, all of the supporting mechanisms that a cytotoxic T cell needs to combat, to find its target and to destroy its target. So you can think of it as sort of a radar that hones in the cytotoxic T cell to its target without needing any other help. CAR is a protein, and proteins are very tricky to deal with. We can't just insert a protein into a cell, but what we can do is teach a cell how to make its own protein. Usually every cell has DNA, and DNA is the main blueprint, main set of instructions for a cell how to make a protein. And there's this thing called the central dogma of biology, and that is every cell has its DNA. That's the booklet that's the entire book of recipes for every protein. The book, a page from a book, a recipe gets transcribed into RNA, so a small piece, a small code, that then gets put into a machinery to make a protein through a process called translation. So you have DNA going into RNA, and RNA instructing this machinery on how to make a protein. DNA and RNA are both nucleic acids. That's where the Na comes from, but they have very different characteristics. So DNA is permanent. It's very persistent. You might have heard of dinosaur DNA or mammoth DNA that we can find from fossils that are millions of years old. It is also able to self-replicate. If you change it, the modifications are permanent. And if you change it, you can cause a mutation that is also permanent. RNA is different. RNA is ephemeral. It degrades very easily. And a lot of work that's been done on RNA vaccines was actually work to make the RNA more permanent than just a few minutes or hours that it's around in the cell. So it has a limited half-life. If you make a change on an RNA molecule, that change goes away with the RNA because the RNA does not self-replicate. It cannot divide itself. However much RNA you put into a cell, that's the amount of RNA that stays there, and there are no changes to the genome. So DNA gets transcribed into RNA, but aside from some special cases, RNA does not go back into DNA. It does not cause permanent changes. And we chose RNA to instruct the cell on how to make a CAR protein. And rCAR-8, which we're testing in myasthenia gravis, is an autologous RNA CAR-T therapy. So first, we discussed what that T part is. It's the T cell, the cytotoxic T cell. CAR in CAR-T is that protein that short circuits the T cell activation mechanism and helps the T cell home in on its target. The RNA is this. The RNA is the instruction on how to make a protein that's less permanent, not self-replicating, that has limited half-life. Especially compared to DNA. And rCAR-8 is also autologous. Now, autologous, I haven't mentioned before. Autologous means it comes from our own bodies. So rCAR-8 is made from the patient's own immune cells through a process called apheresis. Apheresis is similar to plasma exchange that's sometimes used in treatment of myasthenia gravis. So there's a machine that takes some of the blood, it pulls out just the white blood cells and separates them from the rest of the blood, from the red blood cells and the plasma, and those get inserted in the platelets, and those get inserted back, infused back into the patient. So we only get the white blood cells in a separate pack called a leukopack. That's transported to our facilities in Gaithersburg, Maryland. I'm in Gaithersburg right now. Everything's been done here. And here we put the cells in culture, literally in a flask like this, and that's where we grow them from a few 100 million cells into billions and billions of cells. Once there are sufficient cells grown, and that's a process that takes about 7, 10, 11 days, everybody's different, it depends, then we insert the RNA into those billions and billions of cells and freeze them. So those are cells that are now being taught. They were taught how to make the CAR protein, and some of them already have the CAR protein. It starts showing itself very soon after we insert the RNA. We freeze those cells, and we transport them to clinic. We can transport them the next day after freezing, or we can transport them months and months from then when they're needed. So that because they're frozen, they're very stable for a long time inside a freezer. Back in clinic, those cells are thawed and then infused back. And currently we have 6 weekly infusions, and we'll come to how we got to that schedule, being infused once a week. So that's autologous. Now that's this is what they're supposed to do. So myasthenia gravis, as you know, is a complex neurological disorder. Classically, it's thought of as an autoantibody mediated disease. So a rogue plasma cell produces an antibody that targets the neuromuscular junction. Neuromuscular junction is the place where nerve touches a muscle and secretes it secretes proteins, different molecules that help the muscles contract. If this gets destroyed, then your brain wants to move a muscle, but there's no connection between the nervous system and the muscle, and that's what's behind that profound weakness. For antibodies to actually do the destruction, they activate something called the complement system, and this is the large purple stuff you see here. So a lot of drugs recently approved work at this level. They stopped complement from activating. The antibodies are still there. They might get to the neuromuscular junction, but they don't activate the complement. The trouble is we do need complement. We need complement, especially to fight bacteria. So if you inhibit the complement, it can make you prone to bacterial infections in particular. Some other drugs work on depleting the antibody. They don't just deplete specifically the myasthenia antibodies, they deplete every antibody. So and then that causes some suppression of the immune system of its own. And they don't affect the plasma cells. So the plasma cells that produce the antibody are still there. And both of these kinds of drugs require what we call chronic administration. You need to get them frequently for a prolonged time in order for them to exhibit their effect. Now, the plasma cell didn't just pop out of nowhere. It differentiates itself from something called a B cell. And we think that those B cells are also gone bad, quote unquote, and drugs like rituximab work at this level. But again, those plasma cells that are already made, they're there. Rituximab does not hit the plasma cells. And we think that T helper cells are those that prod the B cells to become the plasma cells. So they're also part of the disease process. So Descartes-8, we believe, acts through two separate mechanisms. One is a direct effect. It hits the plasma cell, and that's where the CAR protein comes in. But also they are T cells, and there are tens of billions of T cells that affect the T cell component of the immune system in ways that we're now finding out about, and we're doing some lab work that's still very preliminary, but we think there's also an effect there. So that's a nice story. The thing is, we need to show and we need to see whether it actually works, whether it works as we expect it to. So we have a hypothesis that it would work through this way, and that infusions of Descartes-8 would cause improvements in symptoms of myasthenia. But how do we actually know that? How do we in medicine know that some intervention, whether it's a drug or a procedure like surgery, whether those actually work? And 150, 200 years ago, you had people who were claiming all sorts of things and were able to legally sell anything they wanted without anybody overseeing whether what they're saying was actually true. But through trial and error and through a very long process, we've come to the medicine as a field has come to the clinical trials as a way to test whether some intervention, whether a drug or a procedure is A, safe, and B, effective, whether it's because the primary thing in medicine is do no harm, whether it's safe to administer and whether somebody can tolerate getting it, and two, whether it's doing the job of alleviating symptoms, improving life, prolonging life. And there are various stages of clinical trials from phase 1 to phase 4. In phase 1, when you have a substance that's never been tried before, you give initially a small dose and then an increasing dose to a fairly small number of people, either healthy volunteers or people with a disease that the drug is supposed to target, to establish, one, a maximum tolerated dose, so what's the highest safe dose that you can give, and two, a recommended phase 2 dose, which can be the MTD or sometimes it's lower than the MTD. In phase 2, you're increasing the number of people who are getting the drug or the intervention, and you're testing various schedules in which to give them. And you're also looking at rarer side effects, those that don't show up in every patient or in every other patient, but that they might show up in only 1, 5, 10% of people. You're also looking at initial signs that symptoms are actually improving and that it's worth testing that drug more. In phase 3, you're comparing the drug against a placebo, against a dummy drug, or some alternative treatments if giving placebo would be unethical. That's usually the case with cancer, where there usually are other approved treatments. So it would be unethical to give a patient placebo where they could be getting something that's active against the cancer. This is usually the stage where if you show that the drug is better than the alternative, than the control that you're giving, that's usually when the FDA allows the drug to be marketed, commercially sold. That's what we usually call FDA approval of a drug. And then there's phase 4, that's after the drug is available commercially, where if there are any leftover questions about how the drug works in some other diseases or in a certain smaller population, that's when you do the testing. That's also when you see very, very rare side effects that might show up only if thousands of people have been exposed to the drug. So rCAR-T is now being tested in a phase 2b trial. It went through phase 1, that was part 1 of this study, protocol MG001, where 3 patients with myasthenia got increasing doses of rCAR-T. Small dose one week, and then a medium dose the second week, and then the highest dose on the third week. These doses didn't come out of nowhere. rCAR-T was tested first in patients with multiple myeloma. It's a cancer of the plasma cells, and because rCAR-T targets plasma cells, that was sort of the standard way of testing treatments that target plasma cells. And it was given to more than 30 patients with multiple myeloma without any dose-limiting toxicities, really without any side effects at all that could be attributed to rCAR-T. So the FDA allowed us to test it in a smaller number of patients because we had that prior experience with patients with multiple myeloma. In part 2, we wanted to see how frequently we can and should give the highest dose. So there were 3 separate arms of the trial. Some patients got the highest dose twice a week for 3 weeks for 6 total doses. Other patients got it once a week for 6 weeks, 6 total doses. And then we also had a part of the trial where we gave it once a month for 6 months for 6 total doses. And after 12 patients were treated, we determined that 6 weekly infusions led to deep and durable responses. So the once a week treatment is what we're testing now in a phase 2b trial. It's a randomized controlled trial. So it's not a phase 3 yet, it's 30 people. Phase 3 trials in myasthenia are usually doubled or triple that. But we wanted to test it in a smaller number first because this is the first ever placebo-controlled trial of a CAR-T therapy. That's not only placebo-controlled, it's what's called double-blinded or double-masked, where neither the patient nor the treating neurologist knows what the patient is getting. And I'll come to that in later slides, but first I'd like to talk to you about the part 1 and the part 2. So in part 1 and 2, we were evaluating the safety first and then efficacy of rCAR-T in myasthenia. And the question is, how do you actually evaluate that? Safety is fairly easy. You talk to the patient, examine, and then report whatever new and unusual happens from before the patient started treatment to during and after treatment. So we report the type and the frequency of, we call them adverse events or side effects, if you will. Efficacy is tricky in myasthenia gravis, as some of you or most of you will know. In oncology, it's not simple, but it's straightforward to measure the size of the cancer, of a tumor that you see on a CAT scan. And you can measure how large it is, and you can see if it shrinks and if it grows. And there's a hard number that you give that's a sort of an objective number that a radiologist measures on a CT scan usually. Myasthenia is not like that. Myasthenia, there's muscle weakness, and there's usually muscle strength that needs to be tested by the neurologist. And there are actual symptoms and limitations in their activities of daily living that people experience who have myasthenia. So that's why neurologists came up with these 4 different scores that attempt to quantify what is ultimately a subjective experience. 2 scores are completely subjective. They're what we call PROs, or patient-reported outcomes. The first, and the one that's been the most in use, and the one that actually the FDA recognizes as a valid endpoint in clinical trials, is called MG Activities of Daily Living, or MG-ADL. There are 6 groups of questions, and either patients fill out a questionnaire, or in trials, the neurologist will actually ask. There are guided questions that are being asked and scored. If there are no symptoms whatsoever, everybody would get a 0. So 0 means no symptoms, and myasthenia does not affect activities of daily living at all. And then a score of 3 is the highest score you can get. That means that's the most limitation in that certain area. And there are 8 areas, and the score of 3, so the MG-ADL score goes up to 24. The higher it is, the more myasthenia affects the person's symptoms or the person's lives. Another complete PRO is quality of life. That's has more questions, more points. It's also a bit noisier, in that it varies day to day how somebody's feeling. So it's not really been in use a lot in clinical trials, but we still look out for it. A more objective, quote unquote, way to measure symptoms of myasthenia gravis is the QMG score, or quantitative MG. It actually uses neurologist assessments of how strong some muscles are, along with instruments to measure how hard you can grip your hand and how hard your respiratory muscles work, with an instrument called a spirometer. And that gives you an actual number, a hard number that goes from 0 to 39. And again, the higher the number is, the more severe the symptoms are. Finally, there's MGC, or MG composite, that attempts to combine MG-ADL, a purely subjective score, with QMG, that's sort of a semi-objective score, into this composite score. And that also is the higher the score is, the more severe the symptoms. And there's been consensus among neurologists that a 2-point decrease in ADL and that a 3-point decrease in QMG and MG comp is clinically meaningful. It's more than you would expect from normal day-to-day fluctuations, and it's what patients with myasthenia perceive as an improvement in their symptoms. So we tested DesCART-8, as I said, in a phase 1/2A trial through 2 different parts, and we recently published these results, and you may have heard some of that in the Lancet Neurology 2 months ago now. And when it comes to safety, we didn't see any dose-limiting toxicities. There are 2 side effects in particular that are common in conventional DNA-based CAR-T therapies. They're called cytokine release syndrome and neurotoxicity, and they have to do with the permanent nature of modifications of those conventional CAR-T treatments. So we didn't see any of those side effects, those kinds of side effects with our CAR-T therapy, with DesCART-8. And then those conventional CAR-Ts, again, because of the way they work, they require lymphodepletion chemotherapy. And that's another thing you may have heard about CAR-Ts, because we don't rely on the cells proliferating, because RNA does not replicate as many cells as we put in. That's all that we expect to have, and that we do the ex vivo proliferation in the culture, if you remember. There's no lymphodepletion chemotherapy required, so we did not see any low blood counts, anemia, anything like that. And all of the treatment was actually outpatient. So the conventional CAR-T therapies usually require inpatient admission, that we did not require. And it was all done in outpatient infusion centers. Even more significantly, we actually saw evidence of clinical activity. So here on the top left is the MG-ADL score. the subjective score, if you remember. Here's the QMG score, the more objective score, and the MG composite that combines the two, and quality of life is here. And if you remember, a 2-point decrease of ADL is clinically meaningful, and 3-point decreases in QMG and MG comp are clinically meaningful. Here we plotted out the mean change from right before treatment in every patient. And these are combined scores for the 10 patients who got either twice weekly treatment or once weekly treatment. So everybody had 6 infusions, either over 3 weeks or over 6 weeks. So in those patients who got it over 6 weeks, the treatment ended here, where you see the green arrow in all of the 4 charts. And you can see that during treatment and definitely after treatment, we saw clinically meaningful decreases. They were twofold or even threefold more than what is considered clinically meaningful, actually. And more importantly, we saw that these improvements persisted for long after the treatment ended. So treatment ended here at week 6, and here you see up to month 9, these improvements in scores persisted. And the trial ends at the month 12 time point, and now we have 6 of the 7 patients who received treatment once weekly. They reached month 12, and 5 of the 6 maintained greater than clinically meaningful decrease in all 3 scores at month 12. 1 of the 6 had their symptoms worsen to where they were before treatment. That patient got retreated. The protocol includes an option for retreatment, and they had their scores improve again to what they were after the initial round of treatment. So we're hopeful that people will get at least 12 months worth, some even more, of clinical benefit from 1 6-week round of infusions. So it's one thing to note these scores and their graphs, but what's more, I think, closer to people is to hear a patient's own experience. And we've had one of the patients who actually had seronegative myasthenia tell their story to the MGFA blog, and that story is still up. You can see a link below. That patient has been off study, so they reached the 12-month time point, but we hear, and now they're 16 months. At 16 months, they still had significant improvement in scores. They're a forester by trade who they were planning on early retirement because of the severe symptoms, the diplopia and fatigue, and it's a physically demanding job, so they wanted they plan on selling their business. But they were back working full time and doing, I don't know, 10-mile hikes, 20-mile bike rides after the Descartes treatment. So I'll end with that. I hope that you learned something about how we can use engineered cell therapy to use the body's own immune system to fight even an autoimmune disease, even a disease of the immune system. I hope you understand how RNA CAR-T therapy is different from conventional DNA-based CAR-T therapy and why it has less potential for toxicity. And you saw the results, and you were able to read the results in the Lancet Neurology of the first rCAR-T therapy to be used in any autoimmune disease. And we're happy that myasthenia gravis is the first autoimmune disease that was tested. And this is our 30-patient team. This is in our offices in Gettysburg, Maryland. You can see the site below. University of Kansas is open as a site, and then contact information for University of Kansas is there. So I'll end here, and I'll be happy to answer any questions.
Thank you so much. It's fascinating to me. Really exciting developments for all of us with myasthenia gravis. So we will take questions. You can put them in the chat. And I apologize about the other cameras on the panelists. We somehow have a setting that won't turn on, so you can't see them, but they are here. Do we have any questions? do you guys want to share what other sites are available? We have people tuned in from coast to coast.
Of course. So starting from the Northwest, we have Oregon. So OHSU is a site. In California, we have UCI and San Diego. We have Alberta in Canada. University of Kansas and University of Colorado are open. We will have Tucson open as a site too in Arizona.
Right.
Now Hafsa will help me out with some of the new sites. We'll have Houston open soon, I believe.
Correct.
On East Coast, we have in Florida, we have 3 sites actually, in Orlando, Boca, and Tampa, USF in Tampa, and then two neurology clinics, one in Boca and one in Orlando. We have UNC, Dr. Chip Howard was one of the senior authors on the study. And then we have, is that all? Or am I must be missing some.
We have a site in Pittsburgh.
Yes, Allegheny.
And then we have a site up in Amherst, New York, that Neurological Institute.
And Augusta, Georgia.
And Augusta, and then we'll be also opening up Tufts up in Boston.
Fantastic. Amazing. It looks like we do have some questions that have come into the chat.
So did any of the patients have blood that was not viable for the process? So to date, no. There were, and that's published in the trial, for 3 patients of the initial 14 that we published the data for. The dose was slightly, the number of cells we made were slightly lower than the target dose, but we didn't see any issues in terms of response. So we don't know that the dose that we target is important. So we're still seeing how low can we go in terms of dose, but there was nobody that we actually were not able to make any amount of cells. In terms of cost, that is a long way from now. So we're at the phase 2B stage now. For FDA approval and commercial availability, we need to show at least one phase 3 trial where it's better against the control, and that'll take years. So I with inflation what it is, and with the I wouldn't even be able to guess what the cost could be, because it is a moving target. So given the need to work with the patient's own cells, how intensive is the work required, and what will the cost be like? Again, the cost I hope I've answered. It's probably not a satisfying answer, but it is what it is. And for the scaling, I mean, we are 30 people total in the company. Not an insignificant, but a small portion of that does the manufacturing and other things related to the release, and we are able to produce it for the trial patients. So the benefit of using RNA compared to DNA is that actually you don't need to make DNA. You actually need to work with a virus. Usually it's a lentivirus, and there are a lot of regulations and processes that you need to follow that can be a bottleneck. There's none of that with RNA, and a lot more RNA that we need is being made for, let's say, vaccines. It's a drop in the bucket. What we would need to make to treat every myasthenia patient compared to what's been produced for myasthenia, for COVID mRNA vaccines. So that's in terms of RNA, and in terms of cells, it's actually, I mean, Hafsa has worked in manufacturing for a while before doing clinical operations, so she'd be the better person to tell you, but it's straightforward. And then for people with poor blood veins, is a chest port or a PICC line preferred? So the infusion is actually only 15 to 20 minutes. So for the infusions, it's not preferred. A peripheral line is even better. I mean, ports and and PICC lines. Port not as much, but PICC lines definitely can get infected, and there can be issues with clots. So a peripheral vein is fine, although there are patients who got it through a central line, and that hasn't been an issue. For apheresis, so to get the actual white blood cells, that could be an issue for somebody with poor veins. But on the trial, the initial screening visit includes a vein assessment. And if the site staff determines that the veins are actually not adequate for plasmapheresis or for leukapheresis, then that needs an apheresis catheter, an apheresis port. But that is temporary. That is only for the apheresis, and it's removed on the same day or the next day after apheresis. So the next question is the idea to treat annually based on 12-month results? It's an estimate. It wouldn't be a scheduled 12-month treatment every 12 months. In some people, the clinical benefits may be shorter, so they might need repeat treatment sooner. For some, it might stretch out to a year and a half, 2 years. So it's a fairly small number of patients that we've treated so far. 14 that have been published to date. So that's something that we'll have to see, and we'll find out with time. So I'm 70 when you say years in phase 3. If it's approved, are we talking about 2 to 3 years or a lot more? 2 to 3 years. So it's not half a decade, decades. It's a couple of years.
And then I think there is a question in the Q&A also.
Mm-hmm. So let's see. Previous pharmaceutical teams have included seronegative patients in trials for new drugs, but then not included that indication when submitting for FDA approval. Seronegative patients then face an extremely difficult time getting insurance approval for these medications. How will Cartesian approach this? Well, I can tell you what we're doing in the phase 2b trial. We are including those patients, and in fact, the patient that was shown on the slide was a seronegative patient. So we're really hopeful that once we in the phase 3 trial, we will include the seronegative patient. So I'm hopeful that will actually show the same, if not even better, benefit for seronegative patients as we do with acetylcholine receptor antibody positive patients. So ultimately, that is all dependent on it is dependent on the phase 3 results and what they show. And one of the reasons why we're also really interested in finding out about the indirect mechanism is to see are seronegative patients those who just have an antibody we haven't discovered yet, or is there some indirect pathway to getting myasthenia gravis? And if so, is rCAR-T targeting that pathway as well?
Thank you. Richard, it looks like you have your hand up. I'll allow you to talk if you wanted to. Richard, did you want to ask a question? Okay. I guess not. All right. Any other? Looks like you have another question.
Let's see, that's in the Q&A or
it's in the chat. Well,
will those needing additional dosing get them prior to FDA approval? So we do allow retreatment on the current trial. So patients who got additional doses got them on the trial too. But ultimately, it all depends. Because it depends on what stage we are of approval and whether the trial is still open when somebody needs retreatment. So there are no guarantees there, really. A lot of it is, will the trial continue to be open? And some of it is not really dependent on us.
Are there any other questions?
Oh, I see. Since we can't see you, which one of you in the which one is you in the group photo. Okay, this, I don't know if you can see my cursor, but this is me.
Perfect.
And Hafsa is here.
Thank you for sharing, and I apologize about the setting on the profile.
No worries.
We should know how to fix that, using Zoom for so long, but I don't know. All right, any other questions? All right, well, thank you everybody for tuning in this evening. We really appreciate the information and the updates, and as we learn more, we will definitely share. Thank you.
Thank you, everyone, and thank you for having us.
All right, have a good evening.
Thank you.
Too. Thanks.
Bye-bye.
Bye.